Chapter 3, Basic Principles and Concepts, Immunology and Pathology, Adaptive Immune Responses of Vertebrates, Part 5. I've used this term over the past few mini lectures, inflammation. Let's talk what this inflammation is. Inflammation is a mechanism that our bodies use in allowing for the repair or the removal of things in your tissues. Think about the last time you got a cut. Within hours, the area around the cut may have got a little red, started to swell. You put your hand on it, you can feel it. It's feeling warm. Your initial thought is, oh my God, it's warm, I've got an infection. When in actuality, no. That warmth, that swelling is a natural reaction by your body to do the remodeling required to repair the wound. Same thing if you had an infection. If you had an infection in the skin, under the skin in the dermal layers, bacteria, virus, parasite got in there. The inflammatory response is going to allow for our immune cells to gain access at a better rate, at a better speed, to get serum proteins into that area, the complement, the antibodies, and whatnot. Inflammation is not an immediate thing. And as we'll talk about later in this lecture, if it's an immediate thing, that's a problem. This is going to be a delayed type hypersensitivity. Okay, this is going to be something that's going to take hours to a day or so to start to develop, to completely develop. Because it's going to take time for the cells to get activated. Because remember, we do not want our immune system to go from 0 to 60 as fast as possible, because then it's going to overreact. And again, we'll talk in a few slides about why that's a bad thing. So we want to have a slow ratcheting up of the response. Can just, down here at the bottom, this lower level activation, take care of the problem? If yes, good, then everything stops when it's done. If no, go to the next step, then the next, then the next. The inflammatory response, this delayed, allows for the cells to get to wherever the injury is. Your hand, your chest, somewhere in your abdominal cavity. Going to take time for the cytokines to be released and to diffuse through the tissues. Diffuse through the tissues, allowing for the cells in the area to become activated, to be drained with lymphatics to the nearest lymph nodes so that those cells can get activated, for dendritic cells to get out here to the site and things like that. Again, we do not want this to be immediate. We want this to be a slow progression. We're going to see that the cytokines, such as tumor necrosis factor, TNF-alpha, TNF-beta, they're going to act as cytokines, activators of cells, activator abilities, and they're going to act as chemokines. Chemokines, remember, are the attractants. Woohoo, come here, come here. Woo, 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 woo. Something bad. Come running, come running. That's what the chemokines do. And they do this by, at the site of where they're at the maximal concentration, which means that's probably where the cells are that are releasing them, the immune cells that are releasing them, or maybe the nearby epithelial or endothelial cells that recognize that there's something not self there and start releasing them. So the capillary beds at those sites, near those sites of the highest concentration, those capillary beds are going to start to rework their architecture. Some of the cytokines, some of the chemokines, as well as, in the next few steps, the complement fragments and things like that are going to cause the endothelial cells that are making up the capillary beds to thin out. So instead of being nice, thick, holding in most of the fluid into the blood, so it doesn't leak, so not much of it leaks out. Well, the cytokines, the chemokines, and everything are going to make them thin out, shrink down, and make them also start to separate from each other just a little bit. Keep holding in the red blood cells, but allow fluid to leak out into the surrounding tissues at near the area of the highest concentration of the cytokines and chemokines. That's why the area of the infection, the area of the wound, starts to swell. Fluid leaking out in that area, and it's a good thing. Fluid leaks out, causing the tissues to start to swell. Tissues get a little more separation between the tissues, between the cells, which will allow macrophages, neutrophils, to get in. Because that same signals are telling the capillary beds to thin out and become leaky are also going to tell those same endothelial cells to send up ICAMs and different surface markers that are going to tell monocytes floating by, stop here, exit out here, and as you're exiting out, change your shape, change your gene expression, and become macrophages. More fluid at the site of infection, more fluid at the site of the wound, also means more drainage to the nearby lymph nodes, which means more antigens, more epitopes of things not you get delivered to the B cells and the T cells. See who recognizes it. Get the cellular response started. Or I'm sorry, the adaptive response started. Now sometimes though, the thing, not you, is too big for macrophages to phagocytize and kill. Or it's too big that the eosinophils can only do so much damage. So what you end up with is what is referred to as a granuloma. It starts off as the formation of fibrous, the alteration of the architecture around whatever that thing is, and then that alteration of the architecture, of that tissue architecture, starts to form layers upon layers, and you get what you see right here. If you look here, what you see at the center there is, I believe, I can't remember if that's a larvae or an egg of a parasite. It somehow has gotten into the tissues. But if you look around it, you start to see almost looks like concentric rings of cells. What happened is that whatever that parasitic thing is, larvae, egg, I forgot to look at the figure legend to know what genus species it is. It's sitting there. Macrophages tried to phagocytize it, and it's just bigger than they are. Eosinophils dump stuff on it. Could only do so much damage, can't be cleared. So what's happened now? The macrophages start forming clumping on the surface of whatever that is, that parasite, start clumping on the surface, and they start forming more and more layers. And then those macrophages, they don't die per se, but they go metabolically inert. So they start to form this impenetrable shell around it, layers and layers of cells, just keep forming and forming and forming and forming. The tissue, your tissue that this is happening in, those cells, the epithelial cells, the muscle cells, whatever they are, start to reform in a different way so that they also will begin to form rings and rings of not quite impenetrable, but toughened tissue. So it's no longer epithelial cells that make up, your dermis. It's no longer muscle cells, muscle fibers. It's now something that's new, something different that just happens to be housed in the dermis, housed in a muscle. And you can actually feel it. If it gets large enough, you can feel this small little lump, if you know what you're looking for. That is a granuloma. In the middle of that parasite may still be viable, or given time, it may just age out. We also see that for some parasites, the formation of the granuloma in the musculature is actually a required step in their life cycle, because this will happen in a prey item that a much larger predator, who happens to be the definitive host, will come eat. The granuloma will be digested away in the stomach, releasing the parasite into the small and large intestine. We see with the immediate. Immediate is the problem when you go from 0 to 60 all at once. This is the mast cells, okay? If you have allergies in your sinuses, this is the bane of your existence. Now, under normal conditions, mast cells are great, okay? They release granules of cytokines and signal molecules, primarily histamine, that will upregulate nearby innate immune cells, also triggering the activation of some adaptive cells. All starts with IgE. Okay, IgE binds to something. Hopefully, most of the time, it's going to be something not you. The variable region of the FAB fragment binds to that. The FC portion, remember the constant, the base of the Y, will then bind to an FC receptor on the surface of a mast cell. This will trigger degranulation. All those granules of cytokines and signal molecules just get spewed out. And then they go and they start just activating all the cells they come into contact with. Again, 0 to 60, like that. You get the vasodilation, kind of like we talked about in the last slide. So you get the swelling of the tissues. The problem is that some of the times what you see is not always something good. You get this, as you see right here, this what they call the wheal and flare. Somewhere in the middle of those somewhat concentric circles is going to be the site of the mast cell that came into contact with IgE. Started to dump out its cytokines, its histamines, and all that stuff, and you started to get inflammation. You get this red that seems to spread as a circle, but things happen. So it spreads. The edge, where you see that edge of the circle, that wheel, that is the edge of the diffusion of the signal molecules. As time goes on, it'll get wider and wider and wider as more of those molecules can diffuse. For the normal, that's not a bad thing. It's something that, is not normally considered life-threatening unless it happens, and it happens so quickly, and the response is in places it shouldn't be. You end up with what is called anaphylaxis. Anaphylaxis, instead of that, the degranulation of the mast cells, triggering swelling of the sinuses or that wheal and flare, the inflammatory response actually causes swelling of the trachea, the tissues around the trachea, which cut off your breathing, so you can't breathe. Your throat here starts to swell. And other types of anaphylaxis, it can get to the point where you have head-to-toe inflammatory response and vasodilation, which means you start to lose blood volume like that. And it's not an open wound where you're bleeding out, but it's all the fluid from your blood is now leaking into your tissues. Your heart's going to start to race because it thinks you're losing blood volume, which you are, so it's going to race, and your body starts to vasoconstrict or shut down blood flow to certain organs to try to save the brain, save the heart. Well, then you start to run into issues of kidney failure, liver failure, and once those start to go, they don't generally stop going. The kidneys just start to shut down, and once they shut down, there's no starting them back up. And, well, it can lead to death. If that immediate hypersensitivity leads to an inflammatory response that is so severe in a generalized area that it impinges blood flow and the flow of nutrients and whatnot in and the flow of waste products out, triggers an overreaction by the innate the innate immune responses, you could get localized cellular death, necrosis. Now, if it happens where that necrosis, that dying cells, one cell dying of necrosis, it bursts and it leaks out stuff, leaks out stuff that could be poisonous surrounding cells. Those cells die, and then it spreads and it spreads and it spreads. The next thing you know, you get a pocket of basically dying tissues that is ever widening and widening and getting bigger. By the minute, by the hour, it's getting bigger as more cells come into contact with the necrotic waste, the necrotic effluent and whatnot. They start to die also. If it's under the tissues, if it's in a tissue, under the skin or whatnot, they refer to this as an abscess. If it's at the surface, it's now come to the surface where stuff can now leak out, then it's referred to as an ulcer. This can be a problem. This is when the immune system goes from 0 to 60 and there's no steps in between to act as a regulatory. Do we really need to go further? Do we need to go further?